Hi, my name is Christina Lusick, and this is my presentation on Huntington's disease and the involvement of the HTT gene. First off, the HTT gene and its wild-type function. The HTT gene is found on the short arm of chromosome 4 at position 16.3. It is also known as IT15 or the HD gene for Huntington's disease. When the HTT gene functions correctly and it is not mutated, it codes for a protein in the body known as Huntington. Huntington is a huge protein that contains over 3,000 amino acids and is essential for proper development. Although the Huntington protein is found in many different cells in the body, its concentration is highest in brain tissue, and it provides axonal support in neurons. Huntington supports the transportation of cellular materials, serves in chemical signaling, protects the cell from destruction, and helps with protein binding. The HTT gene mutation. The specific mutation involved in Huntington's disease is known as a CAG nucleotide repeat. So people without the disease have about 10 to 35 CAG repeats in this specific section of gene. The average is about 20 repeats. People with the disease typically have over 35 CAG repeats in the same section of gene. Extension of the CAG repeats can affect the age of onset of the disease. So the amount of CAG repeats is typically correlated with the age of onset. The mutation causes what's known as a gain of function phenotype, and this is because the protein that is formed from the mutation actually takes on its own function. The mutated protein that is produced traps molecules and produces protein aggregates in the brain tissue, specifically in the basal ganglia and cerebral cortex. And the mutated protein and protein aggregates lead to neurodegeneration and behavioral issues that are characteristic of Huntington's disease. On this slide, I included two images that I thought were very helpful. On the left, what we're looking at is chromosome 4, and we have the short arm to the left, and we have the loci of the HTT gene over here. And then what we have over here are the CAG repeats that are producing polyglutamine. Anything over 35 is going to be considered positive for Huntington's disease. People typically have about 20 repeats, but anywhere from 10 to 35 is considered on the normal range. What I found out when I was doing my research for Huntington's disease is that it's considered one of 9 CAG polyglutamine repeat neurodegenerative diseases. So typically when CAG repeats are found in a gene, it usually leads to issues that are neurodegenerative in nature. And on the right side, what we're looking at are protein aggregates that are formed as a result of the mutated HTT gene. So on the left, we have the normal protein functioning correctly in the body. And then to the right, the protein aggregates or buildup of the proteins mixed with these useful molecules that the body can no longer use because they're getting trapped inside the protein, leading to dysfunctional tissue in the brain. And over here we have a pattern of inheritance for Huntington's disease. Huntington's disease facts. It is an autosomal dominant disorder caused by a defect in a single gene, which we know is called a monogenic disease. The age of onset is approximately 30 to 50 years old, and the extension of CAG repeats can affect this age of onset, as we previously discussed. There is typically a slow disease progression, and men, women, and all races can be affected. Currently, about 200,000 people in the United States are at risk for the disease, and about 30,000 people in the United States have Huntington's disease. So this comes out to about 1 in every 10,000 people. A mutation in the HTT gene results in Huntington protein mutation. This dysfunctional protein traps other molecules in the brain and causes clumps of dysfunctional tissue, and this leads to slow progressive neurodegeneration. So just a recap. The risk of transmission from affected parent to child is 50%. You can see that below in the pattern of inheritance for the autosomal dominant disorder. And then genetic counseling and genetic testing to family members of affected individuals may be considered. Genetic testing for the disease does remain controversial. Here we're going to review the signs and symptoms of Huntington's disease. The early symptoms include changes in personality that can manifest as depression, mood swings, trouble concentrating. The person is just not acting like themselves. There's often a lack of impulse control, lack of inhibition. There can be cognitive issues. The person can develop psychiatric disorders. OCD was one of the specific psychiatric disorders that was mentioned, actually including bipolar disorder as well as one of the earlier symptoms of the disease. Weight loss can occur. Uncontrollable muscle movements called tics often occur. This can all lead to speech issues, and as the disease advances, the person can experience dementia. People with Huntington's disease often experience muscle rigidity and contracture, and the signs and symptoms may progress from 10 to 20 years after basically after diagnosis. So it's not a very quick disease typically, except at the end I have presentation before age 20 is called juvenile Huntington's disease, and this often results in a faster progression of the disease. As Huntington's disease advances, pneumonia caused by the person just being sedentary and not being able to move around a lot is often a common complication or end stage. Treatment and genetic therapy. Currently, there's no known cure for Huntington's disease. No medications are available to slow or stop the disease progression. The only FDA-approved treatment for repetitive muscle movements of the disease is known as xenazine or tetrabenazine. Antipsychotics or antidepressants, which are commonly SSRIs, are used to treat irritability and depression in Huntington's disease, and medications are typically aimed at symptom reduction. There are developing genetic therapies for Huntington's disease, and they are targeted at slowing down the progression of the buildup of the mutated proteins in the brain. So two of the therapies that I wanted to list that are being investigated use microRNA, or the other one is called antisense oligonucleotides. So on the left, the microRNA, it's based on natural genes. It's delivered to cells using viral vectors. What it does is it helps to silence the RNA that are creating the dysfunctional proteins. It's kind of like they take the place so that these proteins cannot keep being produced. They would be administered one time to the patient, and they end up blocking or slowing production of the protein. So again, they're slowing down the progression of the disease. On the right, the antisense oligonucleotides, ASOS, they're synthetic, they're designed in a lab. They're delivered to cells using vectors or no carriers at all. They would require multiple doses to the patient. And what it is an RNA or a DNA type of molecule that binds to genes, and it changes the way that they produce proteins. I've actually included a video by the American Society of Gene and Cell Therapy in my presentation. I know we can't watch the entire thing right now, but I'm going to also link to it underneath my presentation. I think it does a really good job of kind of simplifying the two genetic therapies that I just mentioned, using the microRNA and the antisense oligonucleotides to slow the disease progression. So if you guys want to check it out, all you have to do is click the link under my presentation. Finally, I just wanted to include one slide that went over my interests in the HTT gene and Huntington's disease. I have a personal fascination with prions and prion disease. The mutated protein that is produced in Huntington's disease has prion-like properties and effects on the brain. I remember learning in my microbiology course last year about prions and their devastating effects on the human brain and the devastating effects on animal brains as well, but specifically their ability to evade modern autoclaving and sterilization techniques. Heat doesn't necessarily destroy prions. Prions are proteins, so they're not alive per se. So you can't really kill them. And they do have an infectious nature. So the idea of prions in surgical settings, despite all the precautions that we do take, was startling to me. And I think it's something that deserves a little bit more attention. And when I heard about Huntington's disease, I wanted to dig a little bit deeper and check out its parallels with prion disease, and it was pretty interesting. There was also ample information available on the topic, and I also really liked the potentially promising genetic therapies. It gives a lot of hope to those who have either been diagnosed with the disease or who are carriers. The disease was mentioned earlier in our course when we were talking about the ethical dilemmas of genetic testing for diseases with poor prognosis. And then I finally wanted to pose a question. Would you choose to undergo genetic testing and risk finding out that you were a carrier of a serious, potentially untreatable illness? Could the information affect your quality of life, or would the information itself be of higher value than the risk itself? I don't have an answer to this question. It's for everybody to think about individually. But I just wanted to thank everybody for watching my presentation. I've included my references on the next slide. And again, if you want to check out that video on the 2 genetic therapies they're developing for Huntington's disease, I'm going to post it underneath my presentation. Thanks again.